I already introduced myself, so I'm just going to say a little bit about Algrano for those people that don't know the platform. So Algrano is a online ecosystem for roasters to source coffee directly from coffee producers around the world. So we work with local suppliers to set up logistics to make it possible for roasters in Europe to buy small quantities of coffee, large quantities of coffee, and then we consolidate everything to make sure that we can optimize logistics costs and get everything shipped to Europe. And we're trying to invest more in educational content because it's very important for the industry. And I think coffee industry, I guess, I don't know if Lucia agrees with me or not, probably does. We repeat a lot of information that we don't necessarily know the scientific basis behind it. And people, they don't know a lot about, for example, logistics, or they don't know a lot about the physics, the physical aspect of the coffee, but there's also a lot about fermentation, which is a big topic. And the more we work with fermented coffees, I think the more we need to be informed about them. So that's why we wanted to invite Lucia to join us for this today, because she's not only very vocal about this thing and has a very strong opinion about the topic, but she also has the work experience and the scientific background behind it in a way. But I'll stop talking right now. Lucia, please feel free to introduce yourself and tell people what you are teaching us today.
Sure. No, thank you for that, Luisa. And thank you for this opportunity. Like Luisa said, this is something that's very close to my heart, not just fermentation, but speaking about fermentation and being able to share this information. This is like my favorite thing to talk about. And I haven't done, because I live in Colombia in the mountain, I haven't done a webinar like this all year. I have really stayed away from this type of communication because I've been concerned about the internet connection. I've been concerned about, just really getting my bearings here. So she kind of brought me out of semi-retirement on this topic. So I really appreciate that opportunity to get to connect with you guys. And I do have a podcast where I talk about these things. So I do continue to have that communication, but I feel like so much of this is so abstract and so kind of obscure if you didn't take high school biology, that having the visuals is really powerful. And so I'm really excited to share a lot of examples and things that I've drawn, things that I've doodled. So today we're really going to talk about, this is kind of a facetious name, infusion confusion, because it really came from this idea of adulterated coffees, infused coffees. What is fermentation? What is an intrinsic flavor? What is extrinsic? What is sort of something painted on? And I think another important thing to say is that I do have a background in microbiology. I do have a lot of experience in processing, but I also do have a lot of strong opinions, and being able to tease out what is my point of view and what is my philosophy and what is my opinion versus what the science say. And I think too often in this industry, we just kind of put it all together. Someone speaks with a lot of authority, and then you just say they must know what they're talking about. And so I also, apply a critical eye to me as well. I try to be really clear about what is fact, scientific fact, and then what is just my point of view, my opinion. But we could always do a better job of that. So with that, that's sort of my background. I'll talk a little bit more about where I came to my opinions in this presentation, but we're going to get started with a foundation. My goal is to, you guys have a lot of questions. Luisa sent me some really great questions that I do want to answer, but I realize that before we get to those questions, we need to have the same foundation and everybody make sure we're talking about the same thing. So let's get started. This beautiful slide, I just found this artist. Here's another one. Her name is Dasha Plesson. She's a mixed media artist. So she takes this, you can also look her up on Instagram. She's a Russian artist, and she does some growing of microbes in the plates, but also including some, mixed media. So some of this is paint and glitter, and it's just a really cool, beautiful art. And I just found this quote. I love this quote. We all talk about the same things, but we have not yet agreed what it is we are talking about. And that's my hope today, is that once we get through some of these slides, we'll at least have a foundation and say, okay, at least now we're talking about the same thing. And we still may have different opinions, but at least here is our baseline. And then Lionel Robbins said this about economics, which is also kind of a very amorphous topic. So I think fermentation and economics, there's a lot of parallels in that way of like, we think like we all have money, we know what that means. And yet very few of us understand the theory and the technique behind these disciplines.
Lucia,
so today,
can I just interrupt you very, very quickly, and I promise I won't do it again. I just forgot to say that for everyone that has questions, please write them on the chat or on the Q&A on the bottom of the screen. And we will have a Q&A session once Lucia finishes her presentation. If there's a question that's like very, very, very relevant, then we can stop in the middle and go to that question. So you can ask as we go, but we'll focus on the questions at the end.
And also, I do talk really fast, and I'm trying to get through a lot of it. So I'm really glad this is being recorded so that you guys can like stop and watch it again. So don't worry about it. I am going to go through this fast, but it is supposed to be really dense, and it's supposed to give you that, high density of material and things to talk about and things to think about. So the things I really want, I will feel happy, I will feel accomplished if at the end of this presentation, at the end of this conversation, you understand why saying anaerobic fermentation is redundant. That's one of my personal goals. I also want you to have a better understanding of intrinsic flavor and extrinsic flavor. So what's the variety and what is this kind of processing that we're putting on top of it? I want you to have some understanding of microbial precursors. We're going to talk about that, what those flavors mean when we talk about esters and thiols and all of those kind of fruity components. And then we're going to talk a little bit about microbial origins. So what is terroir and where does that come from? So those are my main goals, and that's sort of the framework for today. And this is where we have trouble with that baseline. So most of our favorite foods are fermented. If you like to eat food, if you like breads and alcohols and chocolates, all of these kimchis, there's a million things I didn't draw on here. And because of that, because we enjoy fermented foods and we can be surrounded by them very frequently, I think that we have an idea of what fermentation is. And because we have this exposure and this repetition, but I think very few of us, especially now getting into coffee, have a deep understanding of what that is. It's kind of like the same amorphous, like, yeah, I know what fermented food is. And so the point of the slide is both to say, yes, we're exposed and it's very ubiquitous, but what do we really know? And in this picture, we talk about wine a lot and beer and cheese and chocolate, but coffee has been kind of left out of this conversation. Coffee is kind of a newer, kid on the block in this fermentation conversation. And part of that, and I really like to make this distinction, is that in a way we sort of ignored fermentation for a long time. And now we've got this like huge push of like, coffee's fermented and it's amazing, but also it doesn't need to be fermented. So it's very different from these fermented foods. So even I will say coffee is not a fermented food in the way that cheese is, in the way that beer is, because if you take milk and you don't ferment it, you will never get to cheese. If you take grape juice and you don't ferment it, you will never get to wine. If you take coffee seeds and you don't ferment them, you can still have coffee. This is not an inherent, it doesn't need to happen. It does happen a lot and it makes it better, but it's not necessary. So that's why I don't put coffee in this category, because the fermentation is optional. It's not essential. It doesn't have to happen. So it's just something to keep in context. Like we've ignored fermentation and now like everything's about fermentation. And I feel like we kind of want to pull the dial back a little bit. And I think I've also not helped in that conversation either. So I'm trying to like pull back my work. So should I skip one? No. So this is we're going deep real fast, you guys. The whole point of fermentation, and when we're talking about this, is really a question about energy and electrons. So these are two very, very simplistic, obviously cartoon examples of how plants or microbes, so the one on the right is a coffee seed, but it really should be a leaf. So it should be a leaf. The leaf is what photosynthesizes. The seed doesn't photosynthesize, but just to make it easy for my drawing. And then on the right, we have a little blob of a yeast cell. So what this is trying to show, if I put my cursor, can you guys see my cursor move? Yeah. So in respiration, we call it aerobic respiration because here you can see the oxygen is part of how this mechanism works so that we can get out carbon dioxide. So we get energy from the sun, photosynthesizes, it creates a carbohydrate, creates that glucose, and then oxygen comes in. And so that's why respiration has aerobic in the title. That is an intrinsic quality of it. It's like saying wet water. Water is wet. That's just how you describe it. Contrasting to how yeast gets energy. So again, this is energy, this is ATP. So the glucose molecule comes in, electrons are exchanged, and then it spits out an ethanol, among other things. But the whole point of and showing you this chemistry is just to show you that in respiration, an oxygen is necessary to receive those electrons. In fermentation, oxygen isn't part of this equation at all. Oxygen is not necessary for this energy exchange to happen. So fermentation is anaerobic by definition. And that's different from the environment. You can have this happen in a very aerobic environment, like a dry process or a natural coffee. Or you can have it in a very anaerobic, low. I like to use the word anoxic to kind of distinguish between the environment and like the mechanism. So the reason why saying an anaerobic fermentation is redundant is that all of them are by definition because an electron is not part of it. So when people say, or when people ask me, yeah, but why? Why is it redundant? The answer is really just because of chemistry, because the oxygen electron does not accept the oxygen doesn't accept the electrons and it's not part of it. So it is anaerobic. And now some people say, well, it's being really picky and it's being very like, you know what I mean? It means that I'm fermenting in a low oxygen environment. And that's fine as long as we're again defining our terms and saying, okay, when I say it, this is what I mean. And when you say it, this is what you mean. And I think this is an important distinction because we're both kind of like lay people trying to talk to scientists, or, we're trying to kind of cross discipline, have these conversations, and just being aware that when we're talking about these things, we may not be talking about the same things. And also, I just like to point out that because of this inherent definition in the word fermentation, maybe when you are talking to a scientist, you can sound a little bit dumb instead of sounding, smart and aware as you want to sound. So just keeping that in mind. The other thing I want to give you is a fermentation framework. So there's a lot of things happening. So I like to differentiate between microbial demucilagination, fermentation, and rot and decomposition. And all of these things, I just made this up, but this is how I like to think about it. I think of microbial demucilagination, meaning the microbes remove the mucilage as the function. So that's like the whole umbrella can be microbes are doing this activity versus a machine doing this activity versus an ecopulper doing this activity. And within that, pie, there's a section that's fermentation, meaning I apply that word only to mean intention. Somebody did this on purpose. We are selecting our microbes. We are monitoring the actual fermentation. And then there's this other part that's decomposition or rot. So that's still microbial activity, but when you just do nothing, when you just kind of let the coffee hang out there and aren't paying attention, for me, that lack of, intention, that lack of activity, I call it neglect. That's why for me it's rot. And if you have this little piece in the middle, that I call this kind of preference or opinion. Like this, there's a little bit of wiggle room between someone's fermentation could be somebody else's rot. And I think that's, where we go from science, like these things are very well defined, to more opinion, to more subjective, like this is, beauty is in the eye of the beholder. So this is a way to think about not everything that we talk about is fermentation. It's not the only thing that's happening. And just to, again, put it in its context, put it in its box. And back to this point of why do we ferment? What is the point of fermentation? So I think I've further split that up into two camps. They have the point of fermentation used to be historically, traditionally, a mechanical function, meaning just removing the mucilage. The point of the fermentation was to get the sticky stuff off so we could dry it and roast it. So it was like this thing that you needed to get out of the way. And now we're talking about fermentation as biological, right? It's not just a mechanical function of removing the mucilage. It's like, well, what if we can add flavor? What if we can, further develop this product and make different things. And one is, like I said, one is innovative in the side of like, what can we accomplish? And then the traditional side is more, let's just get this over with, more kind of volume commodity, let's push this through. And a lot of the times we do use the same word for both of these methods. And again, I think that's where a lot of misunderstanding happens, because some people are like, I need the fermentation to be as short as possible because I need this, like, it's like a barrier. It's like a thing in my way and I want to get past it. And other people are saying like, no, let's do 100-hour fermentation. Like, let's like really like marinate in the flavor. And you're both right? It depends on the condition. So some people will say the best fermentation is 100 hours. And some people will say the best fermentation is 0 hours. You're both right. There is no optimum time for fermentation. It's just, what are you trying to accomplish? So this is when you're having this argument or having this conversation, say, where am I standing? Am I standing from the point of view of biological flavor addition, or am I standing from the point of view of mechanical function? And am I talking about fermentation or rot? Or am I talking about microbial demucilagination? So it's sort of trying to place ourselves. Where do we stand when we have these opinions? So where do I stand? I studied at UC Davis, and that's where I got my degree in winemaking. And I worked at Opus One. These are the two wineries that I worked at the longest, at Opus One and at Cliff Lady Vineyards. And this experience gave me this point of view of the environment of where the fermentation is happening. And that, when I think about environment, I really think about on the microscopic level of what are these yeast and bacteria experiencing? What is their oxygen content? What is their temperature? I'm looking at it from like the tiniest point of view. The other thing I want to point out in these pictures is kind of the uniformity of the grapevines compared to a coffee. And maybe this might be a little bit more familiar in Brazil, but very few coffee farms look like this. Very, very uniform. Everything is trellised, everything to the same size. The rows are completely accessible. It's flat so that you can have machines going into the rows. And then on the right, Cliff Lady, the cleanliness, like this is a beautiful, beautiful facility. And actually, there's another picture from the other perspective if you're looking kind of this way, and there's actual beautiful art that was part of the gallery. And they're just very different places to work. So this is where I got my training. This is where I got my most experience. And applying these types of philosophies to coffee is where most of my opinions come from. So now I'm having 14 years of processing, not in coffee. Like I said, this is what my first harvest in Napa in 2007. So I'm pulling grape skins out of a wine tank. And then the 2017 picture, 10 years later, not that different. Same rubber boots, same late night working on a coffee fermentation. So my experience is physically in the mill processing. At first it was wine and grapes, and now it's coffee. But my focus is, how do I get a product from point A to point B? That's where I'm looking at things. I traveled a lot before I settled in Colombia. I was traveling a lot in North America and South America. I did some consulting in Africa. And so my job was to go to a mill spend about a week there and try and help the fermentation, try and help the processing be more efficient and more consistent. I care about consistency. I care about having homogeneity. I think that's one thing that's really misunderstood about my work. I don't want to do special micro lots. I don't want to do, unique little things. I'm trying to do volume, and I'm trying to do it consistently. You can see what's really missing from my experience is Asia. So I hope that's on the horizon. But again, that gives you an idea of why I think the way that I do when I really focus on wash coffees and I focus on this kind of fermentation style. I'm working in very wet tropical places. So when we think about this role of processing, I picked up this quote. I still believe that creation of quality ends the moment you pick a coffee, that every single step afterwards is about preservation and transparency. And then it says, except for the natural process. So I thought this was really interesting because these are words that we're talking about a lot today, preservation and transparency. And this was in 2010. So I thought, a lot of time has passed and since then, and maybe James has changed his mind. So I reached out to him and I told him that I wanted to use this quote And I was asking him, can I use this? Do you still believe this? Even if he doesn't now, at least he did. And it's in a very influential text to say this. So in an email to me, he said, it's probably a fair quote. Naturals have evolved, but they still do paint a particular layer of flavor over any coffee that belongs to the process very evidently. In rare occasions, some naturals don't taste very naturally, but that is so, so rare. And again, this was in 2019. So he may have again changed his mind, but I think this is really interesting to say that this is a very influential person in coffee. And I think a lot of people think this way, that the process is like this extra layer of paint that we're putting on the coffee, and that there's some intrinsic flavor of the varietal that we're hiding or that we're masking, or that maybe we're tricking people into thinking is a different quality. And this is something that, as a processing specialist, I really disagree with. I really don't think that this is the way that we should be thinking about it, even though it is the way a lot of people do think about it. And this is becoming more important because these types of fermentations, mostly, I think we wouldn't be talking about this now if it wasn't for competitions and this idea of fairness. Like, what is fair? Am I being tricked? What am I buying? I think that this type of conversation is important, and it's about time we had it, but I feel like we're not having it in a very sophisticated way because so few people, remember their high school biology class. So not just James talking on the consumers, but producers as well have had this story told to them. So again, this is a very influential coffee processing book, first published in 1963. It says that speed is a desirable factor. It has been well established that coffee does not improve as processing time increases. So this has been hammered into producers' head, that you have to get through this as fast as possible, that the fermentation is a danger zone and you want to get out of it. And so for decades, for generations, these coffee producers have been operating under this assumption that the fermentation is dangerous and we have to get out of here. And then there's this like new wave of, no, let's hang out in the fermentation. Let's have 60, let's have 100 hours. Let's do days of fermentation. And we're surprised that a lot of producers aren't very good at it, that they're very confused by it. And that when we're asking them to explain to us what they're doing, they don't have that foundation. They don't have that history because it's not a cultural history. They've been told the opposite for, since the beginning of their processing careers, at least here in Central and South America. So let's just be, when you're asking a producer to explain what's going on, please remember, they haven't had that much time to figure it out. It's not like the winemakers that have been doing it for 400 years and they have a really solid protocol. We're all just figuring this out. So the other thing I want to talk about is, where does flavor come from? Because this ties into who's got the power. Who's got the power for this flavor? And we know that roasting is a transformation. We know that roasting has flavor, or the flavor is coming from the roasting. And this was kind of, I think, the idea that a lot of people had. Before, I'm talking about like super commodity, like you just get some green coffee and then in my roasting, I will transform it and I am the artist and we're going to get these particular results. So when we're thinking about this, I just want to remember that this introduction of roasting is a transformation. No one's going to disagree with that. But I think it's difficult to talk about transparency when we know we already have a transformation. So I think this term of transparency is maybe a little bit premature, or maybe not the right way to talk about it, because we already have this one transformation. But we know that coffee is not generic. We've known that the process matters. We know that a wash will taste different from a honey, from a natural. When you put this coffee through that machine, you're going to get different outputs. So that idea starts to break down of, oh, do you just need green coffee and we'll transform it later? What we do to the coffee matters. We all accept this. This is a baseline. But now we're getting beyond these kind of like three categories. And where does that flavor come from? So actually what I wanted to show here was like, we're really focusing on the process, meaning a tank and coffee in a tank, meaning a raised bed and, honey, coffee with mucilage on it, or the patio where you can see the skin still on the cherry. So we've really focused on what it looks like the tank, the raised beds, the patio. What we haven't done yet, or what we're only barely starting to do, is focusing on the microbes that live in those environments. So I know this is really tiny writing and you can't see it, but that's why I try to blow up this picture. So what we're seeing here is that this is for a dry process coffee. And it shows that the microbial population changes over time. So what they did, this was a study from 2008. This was in Brazil. And they took days of fermentation. So 0 to 4, 6 to 12, 14 to 18. And then here we have 22 days. So there was a natural that was 22 days on a patio. Oops. And then they took the moisture. So as the percentage was going 40%, 19, 15, and down to 11%. So what they noticed is as time was passing, the moisture was reducing. And as the moisture was reducing, in the middle here, they identified the populations that were on the surface of the coffee during this time. And as the time passed and the moisture reduced, the population was changing. And what you can see over here is that when the pH is high, 7 down to 6, and this is water activity, and you have a lot of water activity, so a lot of moisture, and it's the early days, then the fermentation is 90%, 96% dominated by bacteria. So most of the flavors and most of the activity are going to come from bacteria. But as the pH drops, meaning there's more acidity, because bacteria, as they metabolize, create more acidity. And as the moisture content reduces, then you're going to be taken over by yeast. Yeast have a more developed cellular wall. They're stronger, they're more robust, so they can take tougher conditions. So less moisture and high acid, those are tough conditions. And that's where the yeast are going to thrive and outcompete your bacteria. So it's really important to learn about this, is that even down at 11% moisture, which is where most coffee is dried to, they were still able to find a significant amount of bacteria, yeast that were present, that were on the surface. And this doesn't mean that this is basically showing that you have a 22-day fermentation, that you have a 22-day microbial activity. It's slow. It's not what you're going to see in a tank because it's a very tough condition, but they're still going on. So I just, if there's any producers watching, I just like to remind you to definitely dry your coffee as much as you can, because even at 11, there's still that opportunity for the coffee to change over time without you intending it. So what this shows us is that a wet process coffee, or washed coffees, are generally, because they are wetter, higher moisture, are more bacteria dominated than yeast, unless like me, you add the yeast in. But if you do nothing, the bacteria will be a lot more, that's what will be more present, versus a dry process coffee that will be more yeast dominated for most of the time. So now we know, if we look at this kind of picture again, that our coffee, our green matters, and that it's not just the process and how we do this, if we do it in a tank or if we do it on a patio, but that the microbes that are present in those environments are also contributing to the flavor. So something that I think is really interesting is in a study, they found that sucrose, the sugar in coffee, can vary between 3 to 8%, which seems like not that much. But if you really think about 3 to 8, you're also almost looking at a 3-time, like there are some varieties that have almost 3 times more sugar than others. And this carbohydrate, this sucrose, where does it come from? That's our photosynthesis. That's from the plant's metabolism growing, and then that plant photosynthesizes and it binds those sugars into the seed. So this is one of the questions that I get often, is about, are some processing methods sweeter than others? Like, is a natural process sweeter than a wash process? And I think I have that in the next slide. But to really think about that, sweetness comes from the variety, not so much the process. And then we know we have organic acids, and we know we have amino acids as part of the picture as well, that are part of this transformation. So this is another study that I think is really important, that I love. And it's talking about microbial derived odor active compounds produced during the removal of the fruit mucilage layer, including esters, higher alcohols, aldehydes, and ketones, can be detected in the final coffee product. So what they did is and so some people have asked me, like, how can you tell? Actually, this comes more from like, how can you tell if a coffee's been kind of messed with? How can you tell if a flavor has been added, or if it's from the fermentation, or if it's from the variety? And that they do with GCMS, gas chromatography, mass spectroscopy. And they have kind of these baselines where we'll take and what this study did is they took green coffee, unroasted, and they run it through the spec. And so they saw what molecules were available, kind of what the signature was. And then they took coffees that had green coffee that had been fermented for a long time with different microbes, and then they put that through the specs. These are both unroasted. And then they took those coffees, roasted them, and then put them through the machine so that they could see what was coming from the seed, what was coming from the fermentation, and what was coming from roasting. So scientists are able to parse out where these flavor compounds are coming from. And they know that these microbial precursors from the fermentation, they provide like the building blocks. And what was thought before was that because roasting is so violent, it's so much heat, it's so much energy, that a lot of these precursors are either transformed or destroyed, that the heat would transform them into something else, or that they wouldn't survive. But what this paper showed is that they are able to find precursors in green coffee that survive the roasting process, and then in a brewed cup are also there. So they're not every single molecule in the green coffee is transformed by roasting. Many of them are, and some of them can maintain through that process. So now we have to think about fermentation as also a transformation, but it's preservation, right? That's the oldest method of food preservation. If you have a lot of tomatoes in the spring, you can them so that you can preserve them for later. So fermentation is a way to preserve our foods and keep them for later. So this method, this process by which we remove the mucilage is also another transformation. So we have the transformation of roasting, and we have the transformation of fermentation. So what you're seeing here is a sucrose molecule, which is a glucose attached to a fructose. And so what the yeast will do is it secretes enzymes so that it breaks this bond of the sucrose, because sucrose isn't as available. Most yeast would prefer to metabolize glucose first. Once that's done, then they'll work on the sucrose, and there's very few very specific yeast that will metabolize fructose preferentially. So what it does is it spits out these enzymes, breaks this molecule, eats the glucose, or absorbs, metabolizes the glucose. And from that, it creates alcohol, it creates carbon dioxide, and then all of these flavor precursors, organic acids, amino acids, esters, glycerol, and polysaccharide. Glycerol and polysaccharide are really interesting because those are the compounds that give weight, that give like texture, that you can have something that's very like syrupy and very heavy, and polysaccharides as well. Like there are these molecules that will start to become longer and longer chains, and the longer the chains that you have, the less receptors in your tongue, and so then they'll feel physically heavier in your mouth. So where you can make some of this density, some of this like fullness and body of the coffee through a fermentation. So fermentation isn't just about fruity flavors. You can also develop a lot of body with these yeast fermentations. So the question that I get very frequently is dry process, or why or how is the natural coffee sweeter, or why does it taste sweeter? And this is really difficult because what this shows is that the only way for yeast to survive and to metabolize and to create all of these compounds is to break down the sugar. So yeast will always be depleting sugar. So you are always in a fermentation, you are always running out of sugar. The way like you get into a car, you have less gas this second than you did the second before. So no fermentation will ever make your coffee sweeter, because the only way a fermentation works is by using sugar as its source. So it's depleting, 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 depleting. So why do naturals taste sweeter? So what this shows you is that amino acids are pretty small. We know that those can permeate the boundary. We know that esters can also permeate the boundary. Esters are like really fruity flavors, like when you have a coffee that tastes like tropical fruit or apricots, things like that, like, yeah, papaya, mango, pineapple. Those are the esters. And then RS stands for residual sugar. So these are the glucose and the fructose. But sucrose itself, it does not go through the membrane. It is too large. And not only that, but what we have here, we have water as we're drying the coffee. We have water inside of the seed. And what we're trying to do when we're drying coffee is get the water out of the seed. So the flow of water is generally out. We want water to go out. But sucrose is hygroscopic, meaning sucrose is attracted to the water. So it is like physically impossible. This is where it's like, it's not my opinion, it's physics, where you would have a situation where water would be leaving the seed and sugar could be coming into the seed. That is physically impossible. It would just be like, I don't know, defying gravity, because that's not what sucrose does. It's attracted to the water, so it would be moving out with the water, not in. It cannot move in. So there was a study in 2006, but this myth remains because it tastes sweeter, right? Like, I know it's still there. So why? Okay, so there's a study in 2006 that said that the sucrose in cherry to seed went down, or it was unchanged in wet dry process, but the glucose and the fructose is strongly affected. So this is this residual sugar. So when the sucrose, it's bound, it's unchanged, nothing happens. But when it's split up, then the glucose is able to, what they found here was 90% less. So in these washed coffees where there was a lot of water, where the water was moving out, they had 90% less glucose and fructose. They had 90% less residual sugars. So because again, the water is moving out, the glucose, which is small enough to actually permeate the seed, was drawing out of the seed. So in a dry process, it's not that the sugar is going into the seed. It's not that it's infusing into the seed. It's that it's just not coming out. It's kind of stuck. It's actually bound in there. Whereas in a wash process, all of that water coming out and all of it being available allows that glucose to also leave the seed. But 90% is really significant. So I don't think any of us would say that a washed coffee is 90% less sweet than a natural process. Like, that's absurd. So it's not just the sugar that is giving us this sweetness. There's a lot of other kind of components of sweetness that, a lot of other components in this flavor that give us this impression of sweetness. So earlier I talked about the polysaccharides and the glycerol that the yeast make. So if you have something that is syrupy because it has a lot of polysaccharides, then your brain is like, syrupy must be sweet. Or if you have something that does taste like a papaya or pineapple that has a fruity component and it has the weight, then your mind is like, oh, this is sweet as well, even though it has 90% less glucose than another process. So I think talking about sugar can also be put in that context. Like, it's important. We do want more sweetness, but there are like many roads lead to sweetness. So these are some examples of kind of the landscape today. Of course, we haven't talked about it here, but I'm sure many of you have heard of carbonic maceration, a lactic natural, and then this is like fermented with hops, fermented with yeast, fermented with, all kinds of things. And so all of a sudden our traditional like washed honey and natural, like the world is just like blown up and exploded in terms of like where are these flavors coming from? So with this, these are just things that like I've personally heard of. I'm sure there's many more that have not even made it onto this list. And this is both confusing, but also really exciting, because it means before, all of the power, all of the focus was in roasting, was like, this was the only transformation that we had control over. And if the process matters, and if the microbes matter, and if we know we can get these flavors, then this starts to tip the balance a little bit more of like, we really want to know where this coffee came from and where we have these kind of levers that we can pull. So now flavor isn't just about the processing, it's processing plus microbes. It's what we actually go through and the identity of the microbes that are there. And so here I've just, the same slide of our favorite foods, but really looking at, it's not just the fermentation that gives these products their flavor, but it's the fact that it is an Enococcus oni, or that it's a Saccharomyces, or that it's an Acetobacter, or that it's a Lactobacillus. Like the identity of the microbes really matter in terms of flavor. And I think that's sort of the next step that I don't think that we're talking about enough in coffee. We're talking about fermentation, we're talking about process, but we're really ignoring the identity of the microbes that are there. What bacteria? What yeast? And with this what comes this further information of why anaerobic fermentation is a really simplistic and not a great descriptor about what's going on. Because with microbes, so oxygen availability is a spectrum, meaning here we have Acetobacter, and it is an obligate aerobe. So this is an open test tube, and this is filled with water, with the liquid. And the Acetobacter, because it's an obligate aerobe, it needs air to survive. So even though fermentation is anaerobic, meaning for that process to happen, we don't need oxygen, Acetobacter as a species does need oxygen. So this will only work. That's why it's only here in the surface. So Acetobacter will be in your naturals and in your honey process on the surface. Then we have other microbes that are microaerophiles, meaning they're just a layer below. They just need a little bit of oxygen, but not the full saturation. This is where, and an obligate anaerobe, these guys down here at the bottom of the test tube where there's barely any oxygen, these are less found in coffee and more found like in your gut, like the microbes that are inside of your body that don't require oxygen. And the two main ones that we talk a lot about, Saccharomyces cerevisiae, the yeast, and then we have our lactobacillus. So you can see that both of these occupy the same area of space. They're in the whole test tube. There's some up on the surface, they're in the middle, and they're in the bottom, and these two, but they're for very different reasons. So a Saccharomyces cerevisiae is a facultative anaerobe. So it prefers an anaerobic environment, but if there's oxygen, it can switch its metabolism and it will use both. So it's a very flexible yeast. That's why I really like it. So it can do both. If you don't have oxygen, that's fine, I'll be there. If you have oxygen, that's fine, I can be there too. Lactobacillus, on the other hand, is aerotolerant, meaning it's not that it's flexible and it can like toggle a switch and use both. It's that it doesn't, first, it's not, it doesn't need it. It ignores it completely. So if it's there, if it's not there, it's not that it switches, it's just not part of its world. So it's just kind of like blind to it. So these microbes are very important in coffee, and they can be found in the same places, but for very different reasons. And they have very different mechanisms. So it's really hard to say, oh, I did an anaerobic fermentation, that's why it's lactobacillus, because that's what's there, because a yeast can do that too. So if we're not actively adding the microbes, it's really hard to say what's in a fermentation, because many of these environments will call upon several of these yeast. And in this slide, I separated them out so that you can see, so I can like really illustrate how oxygen matters to them. But all of these are like, you can collapse these and you will have, Acetobacter on top, and then you will have a Saccharomyces, and then you will have a Lactobacillus in there. Like they're all intermingled, they're all in the same place. And that's why they find their little layers. And yeah, you can have a fermentation. That's why when you have a really, really big fermentation, you will get these layers where the top is different from the middle, is different from the bottom, because they will have different amounts of oxygen there, they'll have different temperature, and they attract these different microbes. So really big fermentations are hard to make consistent, because you're literally having like 10 different fermentations in one, depending on how big your vessel is. So where do these microbes come from? So I do get this question a lot about how can we get microbes there? Like, how do I save money? How do I not have to buy them? And what I want you to know is that they're already there. They're all already there. So in this study from 2008, they just took 1 gram, a tiny little gram of pulped coffee, and they plated it out, and they were able to find almost 1 million different types of bacteria, 200 types of yeast, and then fungi were also in there. So everybody is already at the party. It just depends how you're going to change your environment to see who you invite. So you don't need to buy anything. It's already there. So this is the last point. I just noticed the time. This is the last point, but I think it's really important. So do I have to go ahead? Okay. Thank you. So this is where we can start to talk about terroir, and we can talk about what if the microbes are already there, everything is already on the coffee. And if we're introducing, if we're buying something, are we messing this up? Do we have this risk of losing something that's, very unique? And this is a study that I think is super important that I don't know if everybody or as many people in coffee know about it yet. And it was only published in 2017 by Amy Dudley's group. It's out of Washington. And what she was looking at was wine, yeast, and cacao, and trying to map the yeasts that are used in these products. And what she found is these blue dots over here are wine, these green are cacao, and then the red ones up here are coffee. So what's really obvious is that this diversity, so this shows that coffee and cacao have much more diverse yeast populations. There's a lot more of them, and they just are spread all over the map. Whereas wine is like this really tight little cluster. So what they were looking for was an ecological niche, meaning are these yeast populations, are they crop specific? Are they variety specific? Or are they kind of independent of geography? So is it more about what is fermenting or where it's fermenting? So over here, you can see that also wine is a short band, meaning there's a lot less diversity, and coffee and cacao have a much bigger diversity, which is not surprising. It's surprising to see kind of how much, but we know that wine has been cultivated for 9,000 years. So they've had 9,000 years of like really narrowing and figuring out what works and kind of replicating that. Whereas coffee and cacao really haven't had that much time, a couple hundred years. So of course there's a lot more diversity. So I don't know if it's, inherently it's better. I think there's like a little bit of a interpretation that coffee and cacao are superior because they have more diversity, and biological diversity is always a positive thing. We always want more diversity. But I think just kind of taking in that context, like coffee and cacao just haven't had as much time as wine to refine and find that that specificity. And the other thing that I think is important to remember from this is that even though you have like a really small grouping, a really small band of yeast that are found on wine, I think that this really answers a question where a lot of people say, well, if we're all fermenting with the same yeast, isn't all coffee going to taste the same? Are we going to lose this, flavor? And I think, if you drink wine, you would agree with there's so much diversity in wine flavor. Wine flavors from, a Sauvignon Blanc from New Zealand versus a Sauvignon Blanc from Switzerland versus one from Napa. Like these all do taste very different, even though they can be fermented with the exact same yeast. So even a tiny narrow band like yeast, like wine has, can still create a ton of flavor diversity. So I don't think we need to be afraid of losing the distinctness of these places, because we have an example of wine has still a lot of diversity, even with a tiny band of yeast that are available. And not that coffee has to go there, but just let's not be afraid of that. So this is from the same study, and it shows this side is when they mapped coffee, and this is when they mapped cacao. And what this shows is that unlike with wine, coffee and cacao didn't have an ecological niche. It was actually not crop specific. It was place specific. It was country specific. So this is really exciting and really interesting because it meant that each coffee that they tested in the lab had like a fingerprint. So the coffees were sent, blind, and the researchers were just plating them, running them through the machine, and they could say, wow, all of these have a similar signature. And then when they looked, they were like, oh, all of these came from Mexico. All of these came from Guatemala. And so what's wild about that is that Amy's team, they didn't go to these places and like swab the tanks or get samples of fermenting coffee or anything like that. They just got random samples from all over the world of green dried coffee. And it meant that they, like, on your dried green coffee, there is still an entire like fingerprint of where this came from because of the yeast and bacteria that are still on the surface. So I just think that's very wild. And then also remembering back to that other study that I showed you, that if you're not drying down to 11% or below, all of those microbes are still there. So much so that Amy in Washington was able to, extract this fingerprint. So again, what this shows is very interesting, that what they were expecting to find was that they would only find some yeast on coffee and only coffee, and others on cacao and only cacao. And they didn't find that. They found that some of the yeast that were on coffee in can I pick what's that? Colombia. There's one that's on both slides. So in Colombia, the yeast that were fermenting the coffee were also the ones that were fermenting the cacao. So it wasn't crop specific, it was location specific. And this was really surprising to them. They really thought that it would be crop specific. But when they started to look a little bit more as to why this happened, so then you could say, or you can have the argument, well, there's terroir right there. It exists because this only came from this physical place. This country has this fingerprint, this signature. But that's not quite true, because where a lot of this came from is the people that were moving them. So here we just have this very quick little drawing where coffee was native to Africa, but a lot of it is growing in Central and South America, and cacao is native to South America, and then it traveled to Africa. And when these crops were taken from their native environment, the microbes that fermented them didn't travel with them. They didn't bring their specific yeast and bacteria to do their fermentation. They're kind of like sterile at that point. And what's developed in these places are the yeast and bacteria from people, right? So both of these crops are very physically intensive in terms of hand picking and hand pulping and drying and raking. We're constantly touching them. And scientists can map this, and they can say these yeasts were actually moving with these people as they migrated from landing in one country and kind of moving up somewhere else. So in these countries, this bacterial yeast signature is people generated, not physical generated. And that doesn't mean that there isn't terroir. It just means that terroir is much bigger than we think it is. It's not just the country, it's also the immigrant that came to that country. So it follows patterns of human migration. So what this is really showing is that this idea of adulterated coffee, well, we did that 300 years ago when it came out of Ethiopia. It's been adulterated. We took the plant without its microbes, and its microbes are a big part of its signature and a big part of what gives it flavor and a big part of what comes into our cup. So it's just keeping in mind, when are we, there's kind of this arbitrary line to say this is adulterated and this isn't, and to say where are you going to put that line? So thinking about this idea of intrinsic and extrinsic flavor, I do not believe that microbial flavors are painted on. I think that they're integral to coffee's flavor heritage. Like, coffee to me isn't coffee without the fermentation, because it can happen, but that only happens now. Like, a really transparent look happens with an eco-pulper. It happens with a modern machine with a lot of water and pressure and electricity to remove the microbes. So I don't think it's fair. My point of view is just how can this be transparent if it's an integral part of how this crop, this product, came to be into our cups? So this is where it's my opinion. I don't think that there's this layer on top of it. I think that it's very much enmeshed in it, and that the way that we're talking about these things needs a lot more nuance and a lot more kind of delicacy. So I'm hoping, I made it, you guys. I need to take a breath. Okay, so what I think is, I hope is apparent, is that we're lacking a shared truth for coffee fermentation. We're lacking a baseline where some people are standing from the mountain saying this coffee fermentation is a problem and I need to reduce it. And other people are saying this is how I get my signature flavor and I want to maximize it. And we have different goals. So I don't know that we're going to have a shared truth, but that we should at least acknowledge that we don't have one right now. I hope you see now that green coffee precursors affect the roasted flavor, not just as what is transformed and as a baseline for jumping off for the fermentation, but also that they survive the roasting process. And there are some flavors that come from the fermentation or from the varietal that don't get touched by roasting at all and are still found in your cup afterwards. I also see the problem of the marketing focus is on the process and not the populations. And that's why you can have a coffee that is a washed, but can taste like a natural, can have the body of a natural. And we can kind of cross these boundaries. And I don't think that's a trick. I just think that's innovation. I think that's really cool. If we don't have to work so hard, why would we? And just to remember that native yeast did not travel with the coffee plant. So there's no true native fermentation outside of Ethiopia. All the fermentations, even if you're not adding yeast, have been added just by someone else previously. So native fermentations don't really exist scientifically. That's why I like to call them local. They can still be local. They can have a place. They can be from a place. They have a lot to do with the people. But native is like a very loaded word. It's a very value-driven word. And then, when people ask me about competitions, I'm really unqualified to talk about competitions. But my opinion is, I think it's very arbitrary, and that's fine if it's arbitrary. Let's just, acknowledge that it's arbitrary and say, like, this is where I'm drawing the line and this is where I'm, deciding, but being honest about that. And that is my last slide. I'm going to take a water break.
Thank you very much. So this has been amazing. We have the majority of the people still here as well. So, but we only have 2 questions. So I think you either speak really fast and people have been like really struggling to follow up, or people wanted to wait until the end to send their questions. So if you guys, if there's something that you didn't understand, we still have some time. We're going to go a bit beyond our initial estimated time. So just carry on with us, and then we can ask Lucia a few more questions after she drinks a little bit of water. And one thing that I think it was, just before I start on the questions, one thing I think it was very interesting that we talked about before, is that you're talking about adulteration and where we set this line, and the fact that we lack a shared truth for fermentation. Is that you say, like in wine, for example, in wine, there is a lot of, like, there is a shared truth, there is a shared language, there is a protocol, everything's very standardized. And people look up to wine as if it's something coffee should be like, more like wine. But in your opinion, that's not necessarily the case, because wine lacks a lot of innovation. So I think it's very interesting when you say that coffee is a pioneer's crop, like is an innovator's crop. I don't know if you want to elaborate on that a little bit.
Yeah, thanks for bringing that up. I love that part about, I think most outsiders do look up to wine as a luxury product and as an aspirational product for coffee. Having been in wine, having studied it and then made my living that way for several years, I don't think it's a great product to aspire to. And even the concept of terroir, which is so valued in wine, has really held wine back. And that's why, you guys, I made a 3-part, like 3-hour podcast about terroir and wine and as how it relates to coffee. But the reason I wrote that, or I'd made that episode, 3 episodes, is because terroir was such a fixed idea in France that there's so many rules about what varieties can and can't be planted, what you can add or can't add, how much volume, like they restrict how much wine have a place to continue to keep it scarce and to keep it a rare and valued product. And the winemakers in France have really been following the same recipe because it worked. But then what happened was when grapes came to California or Australia, to the New World, the New World had none of those restrictions. They had none of these kind of like baggage of tradition, and they were able to just do whatever they want and make some really great wine, to the point of the 1976 Paris tasting, the Judgment of Paris, where American wines in a blind tasting beat French wines in terms of quality and flavor. So French wine tasters preferred American wines over their French wines. And they were shocked and betrayed. And there was just like such a scandal in 1976 that this happened. But it was because America and the New World was allowed to innovate, and they didn't have that baggage, and they didn't have these like concepts of terroir, and they had great wine. And France was sort of like, oops, like they really stayed behind. And so it's sort of like this double edge of like, yes, it's great for marketing and you have a rich tradition, and that's really helpful for selling a product, but then again, who are you helping? It's like great to sell it, but in terms of innovation and really holding producers back in terms of what they can do. So I just like to bring up that cautionary tale to producers to say, like, don't be so quick to adopt this, because it's kind of like golden handcuffs. It's like, it's really something that should be at least thought critically and challenged a little bit, instead of what I see is just like blind adoption. This is the way we should be. I hit my microphone. This is the way we should be. Everyone should have terroir. Like, terroir is a thing to aspire to. And I'm not saying that it doesn't exist and that it's not something to aspire to. It's just, remember that there are some shadows.
I'm going to go for this was the first question even before you started talking. So I think this person has been waiting for a long time to hear this. Do you know anything about koji for processing, using koji for processing? And I would expand that as well to ask about other weird or novelty fermentation styles that you've been hearing about, or that you've been working with?
Yeah, so I have not personally worked with koji, and I have not tried this type of coffee. I will soon, because there's a sample coming to me. I think many people who know me know Christopher Ferran, and he's a great friend, and he has done this fermentation, and he just recently wrote a blog post about it. So he has a lot more information about the mechanics of it and sort of his recommendations. I am a fermentation minimalist. I like to do things as simply as possible, and koji is a very finicky process where temperature is really, really important. And most coffee mills that I visited around the world are temperature control, at least that precision, is very difficult to do. So I think that it's not going to be a widely accepted process, because it's expensive to get the starter, because it's very, very technically difficult. It's not difficult to do. You just have to put a lot of attention to it and have very strict temperature controls. If it goes above a certain temperature, it dies. If it goes below a certain temperature, it dies. Like, you really have to stay in this very narrow band. And the other thing is, I really like to work with the mucilage and the part. And so what I've seen, and I could be wrong about this, is that a lot of the koji process is done on green coffee, on like when it's already been mostly processed, and it's kind of this further step. So all that to say is I'm cautiously optimistic. I've heard really good things, but I have not personally done it. And I have promised everybody a podcast episode about this soon, and I just haven't gotten around to it, but I will do one.
And there's someone else asking as well, as you come from the wine industry, where fermentation is achieved mainly through added yeast, have you tried fermentation using bacteria inoculation?
So this may be something that people don't really know, but almost all red wine does have a bacterial fermentation. So there's a primary fermentation in wine, which is our yeast alcoholic fermentation. And the secondary fermentation is a lacto fermentation. So it's where we turn malic acid into lactic acid. Malic acid can be very sharp and tart, and so we want to soften that to the lactic acid. They call it a malolactic fermentation. So almost all wine fermentations have a bacterial fermentation where we inoculate with bacteria, and most large commercial wines do. So yeah, I have inoculated with bacteria almost every day in wine.
But in coffee, have you done that already, or?
No, the reason I don't work with bacteria is because what I showed you guys with the environment is that there's millions of microbes present at one time, and you can't be sure unless you have a lab and you can take a swab and plate it. And even then, you're kind of looking at a moving target. And because everything is present all at once, it's hard to know what's there. And that only matters because I care about reproducibility. I want to make 10 containers that taste exactly the same. I don't want to just do a micro lot that's a one-off that's great. So I like to be sure that I can reproduce my results. And that's why I inoculate. It's like my insurance. And yeast, I mentioned earlier, has a more developed cell wall. It's more robust. So you can dry it. You can freeze dry the yeast, and it keeps for a really long time, and you can travel with it. So it's much more accessible. Whereas bacteria, as we showed you in the slides, it needs a lot of moisture. So a lot of bacteria either has to travel in water, it has to travel hydrated, or it has temperature requirements. So when I worked in wine and we did the bacteria inoculations, we would pay, it was like 5 times more expensive to get the bacteria because it would come in a freeze-dried like cold box with ice and the little carbon dioxide packets because it has a much higher temperature requirement and a moisture requirement. So it was a lot more expensive. So it's really difficult to bring with you. It's difficult to scale and it's a lot more expensive. So if you think yeast is expensive, bacteria is way more expensive and way more difficult with the technology that we have today. So that's why I don't use it, because of the scalability and reproducibility. But I think it's great. If you have access to bacteria, you should do it.
Since you're talking about price, this is not necessarily the next question, but there's someone asking about how fermentation affects the cost of a coffee. Have you done any studies on the coffees that you've produced yourself?
Yeah, the yeast that I use is the Lao Cafe brand, and they have the most research behind them in terms of being used for coffee. Not necessarily using a beer yeast or a wine yeast for coffee, but these are been developed for coffee, meaning that they have a much more broad temperature tolerance, so that you can use them in they're more versatile that way. And so with those yeasts that have been developed for coffee, if you can get them at scale, meaning instead of ordering like a 500-gram pack, if you could order a pallet of it, you could get down to 3 cents a pound added cost of the yeast into the coffee. So it's very, very affordable, but it's kind of like this pharmaceutical where like the first pill costs $1 million and the second pill is like 2 cents. It's like that startup cost is really expensive, but afterwards it completely diminishes. And if you can get to scale, it can be 3 cents, 3 or 5 cents a pound. It's very accessible. You just have to get there.
Okay. Going back to something that you mentioned in the beginning, that you wanted people to understand why anaerobic is a redundant word. So we have Max here asking, so first he says that we agree that the environment where fermentation happens changes the taste, because it changes what kind of microbes are active there. Would you still say it's redundant to say that a coffee is fermented in an aerobic environment? I think he's trying to think in a way of like how he can talk about coffee in a way that people still understand what it is, but he's not being redundant in how he's using the words.
Yeah, the way that I like to work around that is to just describe it. Instead of trying to find a label that we know describes something, just describe it. So I would say it was a 37-hour fermentation at 16 Celsius in anoxic conditions. And that way you can talk about the temperature, you can talk about the time duration, and you can talk about the environment, but you're still not necessarily labeling, what was there, because it's very unlikely that you know what microbes were there and that it was a group of many, many microbes. So I think that's where we sort of get into that conversation of, well, what is a description, what is a label, and what is intellectual property? Because you're basically giving away like your recipe. Now you're saying, I fermented it for this many hours at this temperature in this environment, in this type of tank or whatever. So I think that's, it's a workaround and opens up another problem.
Okay. I think we need, maybe you need to explain something a little bit better here. For Cedric, he's saying, I didn't really understand why yeast is integral to coffee. When you're mentioning that yeast were local from one region plus travel with people during the migrations. Is there a timeline there that maybe wasn't very clear? I'm not entirely sure.
So I think this is another thing to just accept, that yeast and bacteria are like their ecological niche are on the skin of fruits and vegetables. So it's not just coffee. It's, whatever cherry or apple or whatever fruit that you have that exists in nature is covered with yeast and bacteria. That is just part of its heritage, as well as humans. Our skin, we talk about our internal microbiome. I think a lot in health are talking about the microbiome, but we also have a skin microbiome. We have all of these yeast and bacteria on our physical surface. And so when, and it's hard to separate. With humans, we have like more bacteria DNA than human DNA. And so we can't separate these two. And that's what I want to use that analogy for coffee as well. So I think a lot of people want to say, oh, the microbes are here and the varietal is here. And I'm saying that it's really mixed in just like humans. Like we can't say I'm here and my bacteria is here. It's like it's in everything and it's everywhere and it's permeated everywhere.
Okay, so I have another long question, but you're not a short person, so I think we're fine with long questions.
I actually am a short person, but I'm a long-winded person. I'm a tiny person.
This is from Dave. He's saying, Lucia, that was amazing. Agreed. I have a question regarding cellular integrity and how it's affected by different fermentations. For example, carbonic maceration tends to give green coffees that are darker and more mottled on the surface. Don't know if I understand it very well here, but so when I'm roasting, then it seems that they have inconsistency in the porosity and the moisture is driven off very quickly from the outer layers. Are these heavily, inverted commas, fermented coffees likely to have outer layers that are more porous?
I haven't thought about it that way. I kind of want to say yes. So I think where this question comes from and what's really the most striking thing in this question is if you're noticing that it's roasting differently, that to me feels like a moisture content issue. And this is my biggest concern with carbonic maceration is that, or dry processed coffees, is that when you have the skin intact, when you have so many layers, you have the skin layer, and then you have the pulp, and then you have the mucilage, and then you have the parchment, and then you have the silver skin, you have all of these layers of tissue, then that moisture that's inside the seed takes a long time to permeate out. And because you're starting with the cherry, all of these different cherries based on their maturity are going to have a different starting initial moisture content. Some may be at 30, they may be too dry, some may be at 50. Like you're going to have this like variability of starting moisture. And now you're starting at all these different points and you're trying to get to something that's consistent. So I don't like that method just for that simple fact of you're introducing a lot of variability, and it's really hard to dry coffee. It's really hard to dry coffee well. And so you're kind of like putting yourself like 20 feet back of the starting line. Like, why would you disadvantage yourself so much when you're already doing a very difficult thing? So that's why I like to work with pulped coffees, because when you pulp and when you remove that outer layer and we can submerge them, then all of my coffees that were starting at these like different moisture contents that are like 30 and 35 and 40 and 50 and all of these different levels, when you submerge them and remove the skin, we can get everybody up to the same level. Maybe you increase the moisture on some of them, but it's like you get to the same level and then you can more homogeneously get down to a very consistent level that would make it easier to have a consistent moisture content and have easier roasting. Also for the carbonic maceration, I do think that it can be a lot of stress, at least in some of the ways that I've seen it in terms of the bag method. You're increasing the pressure. It could be a lot of weight of cherries on top of other cherries. There could be a lot of kind of manhandling, a lot of kind of moving from one place, moving to the other place. There's a lot of physical stress. And I think every time you have some of that physical stress, you are kind of chipping away at the structural integrity of the seed. And every time you're kind of chipping away at that integrity, you're losing that kind of biomass, you're losing some of that density that gives you maybe that longevity. So I'm not surprised. Like I said, I don't work with these coffees on purpose, but I'm not surprised that that's the comment. And that's a very common comment of common feedback about these coffees.
Okay, I'm going to do only one last question because we're way over time. I don't know if I can send you the other questions via email, Lucia, and I don't know.
You might, but I can't promise I won't turn it into a podcast.
Okay, no worries. You can turn into a podcast if you want as well. I think there's a lot of your listeners in the audience. I think I know the answer for this, but I'll let you explain because I think you would make a good point here. So Oliver is asking, how do you know at which point a coffee has enough fermentation? So because there are so many different types of fermentation, especially with different durations from few days to 420 hours of fermentation, like a coffee from Finca La Senda that they have at their roastery.
So I think that the easy kind of throwaway answer would be kind of what I already said, like the beauty's in the eye of the beholder. So good enough fermentation, I would usually say, is whatever you like. But I think a little more than that, because I work with producers, I like to say, how do you know it has enough fermentation? Is when I'm working with my producers is saying what is easy to do and what is easy to replicate. So maybe the only thing that we can handle is a 12-hour fermentation because we only have 4 tanks and more cherries coming in. And it doesn't matter if like the best fermentation is 100 hours, like I only have 4 tanks. And if I can't get more, or if only 5 people showed up to work today and I don't have any more labor, I don't have any more hands, or maybe there's a water shortage, my spring isn't giving me water. So I really kind of reverse engineer a fermentation. And this is another question I get asked, is like, how do I decide? How do you decide what fermentation you should use in a particular place? And I like to start with, what do we have a lot of here? Do we have a lot of water? Do we have so much rain we are drowning in it? Do we have so much physical area? Do we have so much flat area that we can have tons of raised beds? Do we have lots of sunshine? Do we have an inexhaustible workforce? Do we like, are we really close to a road? What are the things that we have here in abundance? And then I design a fermentation around those things to make it easier and to prevent, so like best flavor is almost like my last concern, because I think if we do the other things well, flavor will come. So I never start with flavor. I don't really start with, okay, we want a apricot flavor here and we want to do this. It's like, I'm like, okay, how much water do we have? Let's start from there. So how do you know when you have enough? I think another way to answer that would be, is it easy to wash? Is it easy to work with? Is it, maybe you don't have enough of a long enough fermentation when it's still too sticky and maybe it doesn't dry as quickly, or it gets stuck in the machine, or maybe you start to get some mold growth, something like that. I'm really looking at it from how workable is it, like a physical point of view, not necessarily flavor, because a lot of producers don't even get to that point where they can cup their coffee. It's just more of a, how do we make this easy?
Okay, just one last thing. I just got like a colleague nudging me here to not forget to ask you something about infusion and adulteration as well. So it's in that same thing. We're seeing a lot of coffees with really bold flavors. It's not like something with a hint of something or something that tastes of something, but like something that's like coconut in your face. There's no way around it. It's very strong, it's very powerful. Can that be produced by fermentation, or is that something that, oh, maybe there's something there, maybe there's something added there, maybe the coffee was messed with?
Yeah, so there's definitely, there's been a lot of study and there needs to be more, but there's like 144 different flavor descriptors that have been found from fermentation. So something like coconut, something like butterscotch, something like pineapple and citrus, all of these are able to be made by a fermentation, and microbes are really strong. So just because you have something that is high intensity and like very distinct doesn't mean that it's been messed with. It doesn't mean that it hasn't been, but it's not an indicator of like, this is so marked and this is so crazy, because microbes are crazy and you can get that intensity and that flavor in your fermentation. The only caveat that I've mentioned before is that in all of the studies that they have found, cinnamon has not been something that is microbially derived. I do not know of any microbe that creates that flavor. So just having it there is an indication that it's not microbially derived. It may not necessarily be added. It could be maybe coming from I don't know, it's wood. So maybe it was dried on wood pallets from a cinnamon tree. I don't know, something like that, cinnamon bark. So that's the only one that I don't know of yet, but that doesn't mean that it's necessarily adulterated. It could also mean that scientists just haven't found the microbe that makes that. But as of today, 2021, scientists have not found the microbe for cinnamon, but almost everything else, jasmine, rose, all of those have a microbial source. And in terms of intensity, like what we have now are kind of baseline. So how do you know if something is actually from the fermentation more naturally derived, or if it was infused, kind of added later? Some people would say maybe the content, like how strong is the signal? And all that we have now is some baseline. So someone must have already found it somewhere else. And then we can compare it to that baseline and say, okay, usually the concentrations that are found in nature are 10 ppm, 10 parts per million. And this particular coffee has 100 parts per million. So that could be an indicator to say, all right, we haven't found it in nature, so this could be something that has been messed with. But it also means maybe we haven't found it in nature yet. So I think it's just to take that with a grain of salt, that science hasn't solved all of these problems, and it hasn't Also because there's not that much money in coffee research. So a lot of it is just like underfunded. It's not necessarily that it doesn't exist. It's like no one's really looking. So the easiest way to know if something's been messed with, I think, is what's your relationship with the producer? Like how honest can you be? Do you feel like they're telling you the truth? Do you feel like they're kind of scared and hiding something? Like, I don't know if these problems need to be solved with regulation and with like hard laws. I think, maybe we can just solve them by learning to have better conversations and just communicating and just relationships. So I think that I don't think regulation's missing personally. And again, I have my producer point of view.
No, I think we're ending in a very good line there. I think as well, talking to people is the best way to start looking into these problems.
Ask questions.
Yeah, and we have a lot of questions still coming in, but I'm gonna have to say, people know we can't go on further. I'll send them to Lucia, and you're more than welcome to send your questions to Lucia yourself as well. Lucia, I don't know if you want to like leave a contact email. It's very easy to find you online and to find your podcast, but
yeah, most of the stuff, like I've probably talked about it for hours on the podcast, so that would be the first resource. And then actually, no, I do not take messages or questions on social media. The only questions that I take are from the Patreon. I have a Patreon for the podcast, and those are the only questions that I answer. So if you really, really want to have something answered, you can join the community. And again, either it may have been answered on the podcast, someone else has answered it, but that could be a good resource.
Okay, so that's where you should go then if you have more questions. Lucia, thank you very much. This has been amazing.
Thank you.
Hope everyone enjoyed, and hope you enjoyed it too.
I did. I had a great time. Thanks, everybody.
Okay, have a good day. Bye-bye.
Bye.